Welcome to our next video in our kind of second half of genetics, looking at complex modes of inheritance. This one today is going to be on sex-linked traits. So we're going to practice modeling what does the inheritance of genes that are found on the X and Y chromosomes look like? How can we do Punnett squares with these? And how can we interpret and come up with probabilities in terms of the outcomes? So kind of visualizing where we're at here is kind of this is a nice graphic one, obviously, of a karyotype. Karyotypes are pictures of all of the chromosomes in an individual, and we are looking at a human karyotype right here. I know that because we've got 23 pairs of chromosomes. Now we notice that the first through 22nd chromosome are all genetically identical. So here's my homologous pair for chromosome number 1. One of these would have come from mom and the egg, the other one would have come from dad, the sperm, and we notice that that repeats for 2, 3, 4, all the way up through 22. Now these chromosomes right here, 1 through 22, these are the ones that we've been working with up until now when we've done genetics. Because anything on chromosome 1 through 22, it does not matter if you are a biological male or a biological female. We have the exact same genes in the exact same places for all of these chromosomes. However, where we are going to spend our time today is on this 23rd pair. We know that kind of depending on what you inherit on that 23rd pair is what gives you what we would call the biological sex. So an individual that inherits 2 copies of the X chromosome, so 1 copy of the X chromosome from mom's egg and 1 copy from the X chromosome from dad's sperm, that would make that individual biologically female. An individual who gets 1 copy of the X chromosome from mom's egg, because if we look at biological females are only giving out X's. So here's the X mom would give her son. And then that second, instead of getting an X chromosome in the sperm, if an individual were to get the Y and become XY for that 23rd pair, they're what we would call biologically male. So we're going to be focusing on just these chromosomes right here and looking at the inheritance of genes on them. We want to kind of acknowledge and kind of refer back to some of our terms. So when I was referring to chromosome 1 through 22, and I was saying that genetics we've been doing up until now, it has not mattered whether you're biologically male or biologically female. That is because we've been doing autosomal genetics. We've been looking at any gene that is on those chromosome 1 through 22. And again, those are not impacted by biological sex. Whenever we refer to, or if you hear in a problem that a trait is sex-linked, well, then what you are essentially being told within that kind of hyphenated phrase right there is that we are talking about a gene that is on the X and Y chromosomes, meaning it matters what a person has in terms of those 23rd pair, XX, XY, some other combination. Now, we're going to talk about this first bullet in a second, but typically what we expect to see, and you saw this on our first slide of our karyotype, typically, a female would inherit 2 copies of the X chromosome, 1 copy of the X chromosome from mom, and then 1 from dad and the sperm. And males would inherit XY, 1 copy of the X chromosome from mom and the egg, and a Y chromosome from dad and the sperm. The Y chromosome has very, very few genes on it. So the vast majority of the genetics that you will do at the introductory level, which is where we're living right now in intro bio, we're going to be focusing on X-linked inheritance. So we will almost exclusively look at genes that are on the X chromosome. With X-linked traits, males only have 1 allele. So if you look kind of at our typical sort of genotypes that we have up there for males and females for that 23rd pair, we notice that females, if we're looking at any gene on the X chromosome, are going to be diploid. They have 2 copies of the X chromosome, meaning they're going to have 2 copies of any gene on the X chromosome. One they would have inherited from mom and one they would have inherited from dad. But if we look at a male genotype for that 23rd pair, we notice that males typically only get 1 copy of the X chromosome, meaning whatever is passed on in the egg from mom in terms of genes, that's the only thing they have. They are technically sort of haploid when it comes to any genes on that X chromosome. Now, I did say I want to acknowledge, obviously, when we talk about XX for female and XY for males, we're greatly oversimplifying biological sex. So biological sex obviously has a lot complicated layers to it than just that. So for example, one of the things that we'll look at is there's something on the Y chromosome called the SRY gene. So all embryos, when they begin development as a female. So all of the human embryos are going to develop ovaries, for example, in our chests, and then only in biological males will they descend later on. And it is literally the activation, the transcription and translation of this SRY gene on the Y chromosome that causes that to occur, that it basically kicks off the development of secondary male characteristics. Now, what can happen, since it's just one gene, is that if an individual has a mutation on the SRY gene, that means that if you were to look at their karyotype, they would be XY, but their phenotype, everything biologically about them would be XX. So we see kind of how there can be that sort of inconsistency between what we see in terms of chromosomes and what we see in terms of phenotype. Now, what the SRY gene is actually sort of encouraging cells to do, so how it's able to coordinate all of the development all around the body of secondary male characteristics, is it actually spurs the creation of what are known as androgen hormones. So if you look right here, my middle one is androgen and sensitivity. So this would be where those hormones, so those signals that are basically coordinating to cells, telling them, hey, you should start changing the way you're acting in order to create secondary male characteristics instead of female. Sometimes cells, there's a mutation that causes them to become insensitive to that signal. Basically meaning that that signal saying, hey, you should turn into a male is going on, and the cells basically ignore it and just continue on developing as a female. So again, in this case, you would genotype the individual, they would be XY, but their phenotype would be XX. And then the last one is looking at kind of alterations between these two. So there's a common, not terribly common, but common enough, mutation called non-disjunction. So this is when your chromosomes don't separate, and we'll see this when we look at meiosis more in depth. And so this can cause people to have extra or completely missing X and Y chromosomes. So an example that I have down here at the bottom is an individual that has one X chromosome or an individual with three X chromosomes are both going to appear biologically female. Now, typically, because you are losing or gaining an entire chromosome, there's kind of secondary pathology usually that goes on with these. So you would have other sort of physiological effects from having that extra chromosome or missing it. So again, this is just an acknowledgement that we are oversimplifying by just saying, hey, you have to be XX to be a biological female, you have to be XY to be a biological male. But that's where we're going to kind of move forward with this, just for the sake for learning this skill at the introductory level. One of the things we want to acknowledge right here when we're looking at typical chromosome formation is that males, because they only have that one copy of the X chromosome, do tend to be a lot more susceptible to diseases that are associated with the X chromosome. And I'll pull up a few of those so you can see what I'm talking about. And we can kind of see in this picture why that would be. Well, since biological females are diploid, they essentially end up with a backup copy of their X chromosome. So they might inherit a diseased version of a gene from one parent, but they would have to get another copy of that disease gene from the other parent in order for them to have that disease, to show it in their phenotype. That's pretty unlikely unless we're looking at the case of inbreeding. And we'll talk about how that kind of increases your chances of getting multiple recessive traits in your genotype. But males, we notice, they don't have that backup copy, which means if they inherit a diseased copy from mom, it has to come from mom because that's where they get that X chromosome, they have that. They didn't get a backup copy. So this helps us to explain kind of why certain conditions are more common in males than females. Ones that we can look at for this, so red-green color blindness. So this has to do with the formation of rods and cones in the eyes that allows you to distinguish between the wavelengths of red and green. It is a gene that is found on the X chromosome. So again, if you were to have some sort of mutation, that means that these are not forming properly. That means that you can't distinguish between these wavelengths of light. And so a person with red-green color blindness would not be able to see that it says 29 in the middle of this green circle right here. To them, it would just look like a giant circle of random dots of different sizes. The other one that is really commonly associated with sex-linked inheritance is hemophilia. This one was really common in the royal family. And then again, has to do with the fact that if you have intermarriage and interbreeding, like you had with royals for a very long time in Europe, you have an increased frequency or an increased chance of inheriting two copies of that recessive allele. Hemophilia, you have all these proteins that float around inside your blood. So if there's a damage that occurs to some blood vessel in your body, these proteins will essentially go and kind of clot around where that damage was and prevent bleeding from either into the body cavity or externally. Individuals with hemophilia, they have mutations on some of these clotting factors, these proteins. They're the wrong shape, meaning they can't build those little barriers and they can't stop the bleeding. So hemophilia is a bleeding disorder. It can be very, very severe. And then the last one, I wanted to throw you guys an example of something that's on the Y gene, male pattern hair loss. So that's where you get the balding in the center that kind of moves forward. You get the Friar Tuck moment. And then you end up kind of with just that rim of hair around the edges. This is a trait that's actually on the Y chromosome. So this is something that you would find on there. Even though there's not tons of genes, there are some on the Y chromosome. In terms of how we model this, so how we would show it, Much like we did with codominance, you're going to use capital letters again to represent the chromosomes. But in this case, you're going to use capital X's and Y's to represent the chromosomes, the sex chromosomes from each parent. So if an individual has capital X, that is an indication to you that we are talking about a biological female. And if they have a capital X, capital Y, that is an indication to you that we would be talking a biological male. The allele is going to be represented as a superscript. So that is, again, a letter that sits kind of in the upper right-hand corner of that capital letter. We're going to use the same allele depiction in terms of dominant and recessive and Mendelian inheritance, meaning we're going to go back to, let's use the same letter. Capital means dominant, lowercase of this letter means recessive. But now we're going to see that capital lowercase version acting as a superscript next to my X's and Y's. So let's look at that. We're going to pick on hemophilia. So this is the clotting disorder. Again, it's recessive, meaning that you need to have a lowercase h in that superscript in order to indicate an allele or version of the gene that would actually cause hemophilia, meaning this X with a lowercase h indicates a non-functioning protein. This is a bad recipe. It makes the wrong shaped protein, can't do its job. An X with a capital H represents a non-hemophiliac allele. So this is a recipe for a gene that would be on a chromosome that makes the correct shaped protein, would be able to clot the blood. So we notice that those superscripts to represent hemophilia, no hemophilia, are only on the X chromosome. And that is because this is an X-linked trait, meaning this trait, this gene, is only on the X chromosome. You are still going to include Y chromosomes, but you should not have a superscript on them because this gene is not present on the Y chromosome. You are just going to include a Y on it. We notice that we end up with a large diversity of genotypes and phenotypes in our females. So a female, for example, can be homozygous dominant, and she would be normal, non-hemophiliac. She can be heterozygous, and we notice that we get a new term here. This is when we're talking about a recessive disease. Any individual that's heterozygous for that recessive disease can also be called a carrier. Carrier in terms of genetics means that you yourself do not suffer from the disorder, but you carry the genes, meaning your offspring have a possible chance of inheriting it from you. Hence this idea that you're kind of carrying the gene or carrying it. And then last but not least, a female that does inherit 2 copies of the recessive, so the non-functional clotting protein, would be a hemophiliac. So they would have hemophilia. We notice with males, it's very binary. From mom inside the egg, you either get the recipe for a normal protein, so you can clot your blood, or you get the malfunctioning protein and you have hemophilia. So much less diversity there, again, because there's only one. So let's try this on our own. You're going to do a Punnett square just like normal. It's mostly the notation that's changing here. So using the X and Y's, using the superscripts. So we have a woman who is a carrier for hemophilia, marries a man without hemophilia. What is the probability they will have a daughter with the disorder? What is the probability they will have a son with the disorder? All right, so these can be a little tricky the first time through, and I did make them intentionally. I made you differentiate between daughter and son here because when we look at our Punnett square, you can do a quick check of does the one you made look like the one I made. So I put dad's alleles up top and mom alleles down the side. If you flipped them, we should have no difference in terms of our percentage outcomes. It just might look a little different in terms of what we have for combined alleles in here. So dad, we know, does not have hemophilia, meaning he's easy to tell in terms of his genotype. The one X chromosome he has to have that capital H, meaning the non-hemophiliac allele, and then his Y chromosome gets nothing because, again, the Y chromosome does not have this gene on it. Mom is a little bit different. As a carrier, that means she's heterozygous. She has 1 copy of the dominant, meaning she would not have hemophilia. She would present as wild type or non-diseased, but she does have that recessive allele. We then do our normal combination, keeping the same rules that we did like when we did blood type for codominance. The capital letter and the superscript are 1 unit, representing 1 allele. You cannot break them up. Don't do the goofy thing where you do H, where you do big X, and then you do HH. No, no, no. So we look, and there's a 25% chance that we get a female that inherits 2 copies of the dominant. So she would be homozygous dominant. She got dad's copy of the dominant, mom's copy of the dominant. We then have a 25% chance overall that we get a female who's heterozygous. So she got dad's dominant copy and mom's recessive copy. So if we look right here, this half of my Punnett square shows they're females. So the first question was, what is the chance they have a daughter with the disorder? Well, if I look right here, any daughter they have is not going to have hemophilia. So their chance for having a daughter with it is 0 out of 2 or 0%. If I look over at the males, so where we combine one of mom's X's with dad's Y chromosome, I notice that there is a 1 in 4 chance overall in my combinations that we get that X from mom combining with dad's Y, but it is the non-diseased causing one. And then this one down here shows mom diseased version of the gene combining with dad's Y. So that means this half of my Punnett square shows me sons. There is a 0.5% chance. So if I look out of the 2 that show me sons, 1 out of 2 of those does show a male that would have hemophilia. So there's a 1 out of 2 or 50% chance their son would have the disorder. Now, I intentionally said their son or their daughter because I am telling you, hey, don't give me the probability that they have a daughter and that she has it. I'm saying they have a daughter. What is the chance that she has it? Or they have a son. What is the chance he has it? Meaning you need to interpret these boxes in here and say, okay, if we're asking about daughters, I got to find the boxes that only have XX. If we're asking about sons, I got to find the boxes that only have XY. Now, if I were to change this problem entirely and say, well, what is the chance they have a child with hemophilia? What would you say? Good. So hopefully you realize that if we're saying a child or an offspring or something, that's not gendered. It's not referring to biological sex. So then, hey, it doesn't matter whether we're talking about XX or XY. You just need to look at who would be affected. So in that case, it would be 1 out of 4 or 25%. You guys are going to keep the same notation. Obviously, the letter you're using might change a little bit, but you guys are going to go through and do some practice on this right now on page 26. Shoot me an email. Let me know if you have any questions. Good luck.